guys, today we'll go through part 3 of the chapter on the brain for Anatomy and Physiology 1. Okay, we left off just getting done with the brainstem and the diencephalon, and now we're going to go through the cerebellum. The cerebellum, remember, is the little region at the back of the brain. So we have this large cerebrum on the top. In the center of the brain is the diencephalon, and then the brainstem, and then the cerebellum is this area at the back and bottom of the brain right here. The surface of the cerebellum is highly folded, just like the surface of the cerebrum is. The surface of the cerebrum has lots of folds on it to increase the surface area. Well, the same thing happens with the cerebellum. The cerebellum back here has a highly folded neural cortex on the outside or the surface of the cerebellum. The folds that are present on the cerebellum are referred to as folia. The reason that the surface of the cerebellum, the gray matter that's present there, is folded like this, the reason that there are folia present is to increase the surface area of the cerebellar cortex, which, remember, is the gray matter that's present on the superficial portions of the cerebellum. That's where all of the neuron cell bodies are located. The cerebellum is divided into an anterior and a posterior lobe. So when you look at the cerebellum, kind of like this, there's something called a primary fissure. So just kind of a deeper fissure called the primary fissure, and that separates the cerebellum into an anterior lobe and posterior lobe. This primary fissure is closer to the front of the cerebellum, so the anterior lobes and posterior lobes are not equal. The anterior lobe is actually smaller than the posterior lobe is. We can also divide the cerebellum up into two sides called cerebellar hemispheres. Just like in the brain has two cerebral hemispheres, right? The left hemisphere and the right hemisphere. Well, we also have a left hemisphere and a right hemisphere in the cerebellum, right? Since it's the cerebellum, we refer to them as cerebellar hemispheres, right? And hemi is half, so it's like half of the sphere, right? A left half and a right half. The cerebellar hemispheres are, this is separated by the vermis, but really it would be more accurate to say that they're connected by the vermis. So we'll have one of the hemispheres right here, another hemisphere right here, and then in between the two, we have this band of cortex right here, right, that kind of connects the two together, the two hemispheres to each other. So we'll change this separated to connected. The two hemispheres are connected to each other at their midline by the vermis. Vermis is like a little worm, like the appendix. Remember, the appendix is called the vermiform appendix, and vermiform means worm-like. It looks like a worm, a vermis referring to a worm, because this little, the vermis looks like a worm. It's like a worm of cortex right here that connects the two cerebellar hemispheres to each other. When we take the cerebellum and we section it, right? So say we cut it in half and we look at it like from the inside, we can see that the outer region of the cerebellum is gray matter. And then on the inside, deeper inside the cerebellum, we have white matter, right? Just like we saw with the cerebrum, the gray matter's on the outside, the white matter's on the inside. The outer area of the cerebellum that's made of gray matter is referred to as the cerebellar cortex, right? So the cerebellar cortex is the outer, more superficial region of the cerebellum. And then deeper within the cerebellum is the white matter of the cerebellum. And this white matter is referred to as the arbor vitae. We'll talk about the arbor vitae in just a second. We'll talk about the cortex first. So the cerebellar cortex is the outer layer on the outside of the cerebellum. And again, this is gray matter. What gray matter is this is where all of the neuron cell bodies are located. And remember that white matter is where the axons of the neurons are located. The cerebellar cortex is made mostly of a bunch of large neurons, really specialized cells that are called Purkinje cells. These Purkinje cells of the cerebellar cortex are really big, really highly branched cells. Their dendrites have so many branches in them that one single Purkinje cell can receive input from up to 200,000 other neurons. Okay, so remember the dendrites are where messages are coming into the neuron. Okay, so your Purkinje cell, the cell body's like this, right? And then the axon goes off. And the dendrites over here are where the messages are coming into this neuron. These dendrites are so branched that we can have 200,000 synapses, 200,000 messages can be coming into this one cell at a time. And the Purkinje cell processes these, right? It gets all of this input at the same time, and then it decides whether it should send a message down its axon or whether it should not, whether it should be inhibited. So there's a lot of processing going on in these Purkinje cells on the outside or the cortex of the cerebellum. If we look deep to the cortex, so deeper within the cerebellum, that's where we see the white matter, all of these axons that are bringing messages in and out of the cerebellum. The white matter in the cerebellum is organized into a really neat structure or pattern that we call the arbor vitae. Arbor as in tree, vitae, think like vital, right, life. So arbor vitae is the tree of life. It looks just like a tree. When you cut the cerebellum open and look inside it, there's like this white tree with a big trunk going in, and then it branches and it branches out again and again and again, and it ends up looking just like a tree. This is the internal white matter of the cerebellum, where we have axons bringing messages in and out of the cerebellum. This white matter of the arbor vitae relays information to and from the Purkinje cells. So let's just show what this would look like, and then we'll see a picture as well. But if you can imagine that, say this is the cerebellum, and out here we have all of the gray matter, right? This is where all of the Purkinje cells are that get information, process it, and then send signals out. And then in the center, this is where we have our arbor vitae, right? Our tree of life that comes and it branches so that it can go out to all of the different areas in the cortex. Again, what happens is messages come in via these axons of the arbor vitae, and they'll make their way out to the cortex where the Purkinje cells process that information. Right? There's multiple steps and synapses that are involved here, but the information gets processed for our purposes, and then it gets sent back out. Some sort of a command or message gets sent out via the white matter of the arbor vitae. Right, so the white matter is just a transportation system that brings messages out to the cortex and then brings them back out of the, from the cortex back out of the cerebellum again. Okay, so here we see the cerebellum looks kind of like a clam from this view or this angle. It's highly folded, right? So we're looking at it from the outside. So we're looking at all of the folds of the cerebellar cortex where the gray matter is on the outside. We can't see the white matter from this view because the white matter is deeper within the cerebellum. All of these folds, again, we refer to as folia. When we look at the cerebrum, the larger part of our brain, those folds are gyri, right? The gyri are the raised portions, and then the sulci are the little grooves. The cerebellum, they're a lot smaller. We've got lots of little folds. We refer to those as folia. If you look horizontally, right, going horizontally across the cerebellum, we see the primary fissure. That's just a little bit deeper of a fissure that we see right here that separates the cerebellum into the anterior lobe, which is shaded in blue, and the posterior lobe, which is shaded in brown. This is really difficult to distinguish in reality. The vermis, however, is pretty easy to see. The vermis, you guys see right here, going from the front to the back, separating the cerebellum into the two halves or hemispheres. So we have the left cerebellar hemisphere over here, this whole left side, and then the right cerebellar hemisphere is the whole right side. And then again, the vermis just connects them in the center. This is reality. It looks kind of like hamburger meat, kind of gross. It's lunchtime for me. I just kind of ruined my appetite. But this is reality. This is what it looks like. Again, the primary fissure is really difficult to make out. It is like right here. It's really difficult. I can't even tell you where it goes on the other side. I think it comes, no, it comes back here. It goes like this. It's really difficult to make out in reality, but there is a kind of small anterior lobe in the front and then a larger posterior lobe in the back. The vermis is easier to make out. This would be the vermis right here, connecting the two hemispheres, the left hemisphere, right, and the right hemisphere on the right. And of course, the folds are folia. Now we'll talk about the functions of the cerebellum. A lot of the functions of the cerebellum have to do with movement and coordinating the movements that our body makes. The cerebellum adjusts the postural muscles of the body subconsciously to keep us balanced, and this isn't something that you generally think about throughout the day, but the cerebellum is constantly doing this, regardless of whether you're thinking about it or not, so that we can help keep balance. The cerebellum is also highly important in coordinating muscle contractions so that the body moves as we intend it to move. In general, what the cerebellum does is it monitors what we want our movements to look like and what our movements actually look like. And it compares the two to see, did we actually move the way we wanted to? And if not, it helps to send information to the cerebrum so the cerebrum can correct movements the next time we do it. Because of that, the cerebellum is really important in learned movements. So movements that we get better at as we do them over and over and over again. So doing a cartwheel, hitting a baseball, even walking. I mean, these are things that we're not great at the first time we do them. But every time we try, we get a little bit better and a little bit better and a little bit better. The cerebellum is why we do that, why we get better at learned body movements. So the way that it does this, the way that the cerebellum coordinates repetitive body movements is one, it receives information from the cerebrum about the intended movement. We refer to this as the intent. So the cerebellum doesn't initiate the body movements. It doesn't send the first commands out to the muscles to say contract. The cerebrum does this, right? We'll talk about this a little bit later in the lecture, but in the cerebrum, so up here in the brain, we have the primary motor cortex and the premotor area. Those areas plan the body movement and send the commands out to the muscles to say contract. When they do that, a copy of that plan also goes back to the cerebrum. So the cerebrum has the plans, right? It's got the blueprint. Oh, okay, this is what I want to do. I want to swing the bat and hit the ball. That's the intent, right? So it says, all right, this is the intention. Then the cerebellum also receives information from sensory information from the body to see what actually happened with the movement. So it receives information from the eyes, right? So you see what the movement actually looks like. From the inner ear, The inner ear is where we have balance, right? So what did that movement feel like? Did I feel nice and balanced, or did I feel like super wobbly while I was doing this movement? From the muscles and joints, right? Because your muscles and joints, that's how you know the position of your body. You can close your eyes and move, and I can feel where I am right now. I know that my arms are up like this, whether my eyes are closed or not. That's so gross. I did that and I see my arm jiggle. I'm officially like the old teachers that I had in high school with jiggly arms. I'll wear a long sleeve shirt next time. I'm at home, so don't mind the tank top. Anyhow, even with your eyes closed, you can feel your muscles, your joints, your bones. You can feel your position. Proprioception allows you to feel your position in the world, where you are. So the cerebellum receives all of this sensory information from all different places in the body, and it says, what kind of movement did I do? Did I do a great cartwheel? Did I, swing the bat and hit the ball perfectly or not? And then the cerebellum compares these two things. It compares the intent, what did I plan to do, with the performance. What did I actually do? And the cerebellum, when it compares these two things, it says, okay, do they match? Did I do what I wanted? Or was there some sort of a discrepancy? Did I mess up in some way? If you messed up in some way, the cerebellum says, all right, I see, whatever it is, the deltoid contracted too fast and too hard, and you overshot because of that. And it sends that information up to your cerebrum. Because remember, your cerebrum up here, your motor areas, they're the ones that actually plan the contraction and send the signal out to the body. So the cerebellum will send them a signal and say, hey, look, this is where you messed up. Next time you send the plan out, you should change it in such and such way. Then the next time you plan that movement and send these signals out to your muscles, you'll be sending out a better plan. And then hopefully the movement will go according to plan. It'll go more like what you wanted to happen. Hence, our movements get better and better and better as we practice them. And as the cerebellum is able to adjust those movements little by little by little, so that the performance ends up looking more like the intent. That's the classic function of the cerebellum. When you say cerebellum, I think movement. More recently, we are figuring out that the cerebellum does have some other functions that are different or that aren't related to movement. So we're finding out that there's more to the cerebellum than we initially thought. We know now, or we're just learning now, that the cerebellum has a role in evaluating certain sensory input. Certain sensory input like pitch. So people who are really good at music, their cerebellum is highly developed and they're really good at comparing the pitch of different noises or different sounds. Or the like sounding of two words, like when two words sound alike, or two words that sound alike, are they different? Being able to evaluate the fact, no, those are two different words that you're saying. The cerebellum is also used to keep time. So when like tapping out a beat, right? So predicting when the next beat is going to come, or when you're listening to music, tapping along with the beat of the music. Predicting where a moving object is going to be next is another timekeeping task. So when a ball is thrown, being able to predict where it's going to land, where it's going to go next, is in some ways related to the cerebellum. The cerebellum also does have some role in emotions and in impulse control. So for example, in patients who have ADHD, right, attention deficit hyperactivity disorder, there's a lot of impulse control problems. It's very difficult to control those impulses to get up and bang and grab something and do something. And we see that these patients typically have a smaller cerebellum than other patients do. And so we think that there's some sort of a relation there, and that the cerebellum isn't fully developed, so it's not able to give that impulse controllability like it typically would. All right, we'll change gears from the cerebellum and talk about the limbic system. The limbic system is also referred to as the emotional brain or the motivational system for the self, right, the person. The limbic system includes numerous structures in the brain, including what we refer to as the limbic lobe, which is located deep in the cerebrum, multiple cerebral nuclei that are scattered throughout the cerebrum. Two very important are the amygdala and the hippocampus. Regions of the thalamus and hypothalamus are involved in the limbic system, as well as the reticular formation, which is a structure that stretches through parts of the brainstem. Now, these structures that are in the limbic system or part of the limbic system are present in both hemispheres of the brain, so both the left hemisphere and the right hemisphere. And then you can see through this, right, that it includes things in the cerebrum, right, both hemispheres of the cerebrum, in the diencephalon, throughout the brainstem. Right? So there are areas throughout the brain that are all involved in this limbic system or emotional brain. The function of the limbic system obviously has to do with the emotion or our emotions. All of these components work together to establish our emotional states, right, or how we feel. Also how we react to those feelings, right, or how we show those feelings. And all people do this a little bit differently, right? When you get really angry, do you explode and throw things and lash out? Or do you just shut down and pull everything within yourself and hold it all in, go quiet? When you're sad, do you tend to, cry and really express those emotions? Or again, do you just build up a wall and pretend like you don't feel anything? Everybody is a little bit different, and all of this has to do with the way that the limbic system functions. Now, a lot of how we respond to our emotions is learned, right? These are learned behaviors that we learn either from watching other people or from past experiences. You react a certain way and then the world does something. If it's something you like, you'll probably react that way again. If the response you get to that reaction is negative, you probably won't react that way again. Because of this, because our emotions and the way we show our emotions is a learned behavior, it makes sense that these areas in the brain are also involved with or responsible for processing memories, right? Hence, you remember how somebody responded. You remember certain situations and the emotions associated with them. And that's how you know how to express those emotions in future situations. The limbic system is also responsible for providing motivational drives. For example, in the limbic system, we have a gratification center and an aversion center. And this is related to what I just said before. If the response is something you like, if it feels good, you're likely to repeat that behavior. When the gratification center is stimulated, when that part of the brain lights up and starts firing, the person tends to feel good things, right? So the person tends to feel pleasure or a sense of reward. Hence, they're more likely to do whatever behavior it was that stimulated this gratification center because it felt good when they did that. The aversion center is the opposite. When the aversion center is stimulated, it stimulates feelings of fear or sadness. Okay, these are not generally positive feelings, right? These are negative feelings. So the person is more likely to avoid whatever it was that caused this aversion center to be stimulated, right? Hence, it's called aversion. You avert from whatever it is that caused those feelings to occur. Again, I mentioned before that the limbic system is also involved in memory, right? So how we store our memories and then also how we retrieve them at a later date, which we'll talk about in a minute. Here we see different parts of the limbic system or the emotional brain. This is showing us the deep within the brain, right? So all of this out here, this is all the cerebrum, the superficial portions of the brain. This is the diencephalon, right? And then it kind of goes down to form the brain stem. So let me erase those scribbles. Here you see the limbic lobe, which is this kind of whole curved area here that's deep in the cerebrum. So it's in the deeper portions of the cerebrum. You also see that we have parts of the hypothalamus involved, right? The hypothalamus is right down here. Parts of the thalamus, specifically the anterior nucleus of the thalamus, which is in orange right there. We have multiple nuclei, hypothalamic nuclei. These are little masses in the hypothalamus. We said that two of the most important nuclei that were present in the cerebrum were the amygdala and the hippocampus. The amygdala you see right here, right, or amygdaloid body, and then the hippocampus you see right here. Okay, so these are some of the most important portions of the limbic system. There are other areas that are also involved with the limbic system, like you see the corpus callosum and the fornix, which are areas where, like white matter areas where messages are sent back and forth. But I put the most important areas in red. So we call the limbic system the emotional brain, and we said that it is important in emotions, right? It allows us to have emotions, be aware of those emotions, and then decide how it is we're going to show those emotions. But we also said that the limbic system was important in memory, both memory storage and retrieval. Now, memory involves more than just this basic, like having access to your past. In order to form a memory in the first place, we have to acquire new information, and we call that learning. So really the first step in memory is learning, right? Acquiring information from what's happening around you and compiling that into a unified sequence, right? Something that makes sense. Then we can actually file it away. And then we have access to it. We can take it out, retrieve it, and remember it. But learning, right, acquiring new information is really the first step in memory. And then again, the storage of that information and being able to retrieve it at a later date, right? Pull it out of storage and actually remember it at a later date. The information that we're experiencing at any given time gets compiled again into one nice unified memory. But we experience all sorts of stuff, right? We have tons of experiences that we have happening all the time. And we don't actually need to store all of those experiences in long-term storage. We don't need to have access to absolutely everything that happens to us, everything we see. There's a lot that happens that we don't need to know, we don't need to keep. So forgetting unnecessary information is a really important part of memory. For example, like right now, I could think that it's important to file away exactly how much water I have in this right now, and the angle that my phone is sitting at, and exactly the temperature right now, and the fact that I have a red pen and a purple pen sitting here, and then my glasses case is open, and all this useless stuff, right? None of that actually matters. I don't need to remember that. All I need to remember from what's happening right now is that I did record this lecture and that I did upload it. That's what's important for me to remember, so that I know I got it done. That's it. I don't need the rest of these details. So forgetting them, not taking those details, not noticing them, not filing them away, is really important. And the reason for that is that if every detail got filed away into long-term storage, then the person would have no idea what's actually important and what's not, and they would have a really hard time actually processing information. So if tomorrow, someone asked me, did you post that lecture yesterday? I might not know. I might start rifling off all this information without actually knowing the key important feature that, yes, the lecture got posted. I might tell you at 1:22 p.m. I was sitting at the table at this exact angle and blah, 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 and I could list everything that was sitting on my table right now. Useless. But I couldn't actually tell you the important point that, yes, I made the lecture. So you might think, oh, it would be great to have this, a memory where you don't forget anything, but it's not. It actually impairs your ability to think logically because everything is important. So you have no ability to distinguish between information and pull out just the important stuff. You just have this random mass of information and no ability to use it appropriately. So in order to have a good functioning memory, you have to be able to learn information, file it away, leave it in storage for long term, forget unuseful information, unnecessary information, and pull things out of storage. That involves numerous different parts of the brain all working together. I mentioned that the hippocampus was a really important part of the limbic system. The hippocampus, which is remember, it's like a specific little nucleus that's present in the cerebrum, and then also other areas of the cerebral cortex or the general cerebral cortex are both really important. And these two things work together in storing and retrieving memories or information. So when we talk about the hippocampus is what I just kind of nickname the fast learner. The hippocampus is a fast learner. The rest of the cerebral cortex, right, this mass of the cerebral cortex, is a slow learner. So because the hippocampus is such a fast learner, it's able to organize all this incoming sensory information as it happens. So as you're experiencing something, the hippocampus takes all that sensory info and it organizes it into a nice unified memory. So the hippocampus can do this really fast as something is happening. But the hippocampus forgets things really quickly. So we can't rely on just the hippocampus. Otherwise, when you woke up, if you wake up tomorrow or the next day, you won't actually have access to that memory. You'll lose it. The hippocampus is really quick, but it's short term. So what happens is the hippocampus has to file away this information. It has to pass it on to a long-term storage facility. That long-term storage facility is the cerebral cortex, right? They're the limbic system portions of the cerebral cortex. Now, the cerebral cortex is a slow learner, so it's not fast enough to pick up the information as it happens. However, it is really good at storing things long-term. So what happens is, after the hippocampus creates a nice unified memory, it uploads that memory to the cerebral cortex over and over and over again. And the cerebral cortex is able to pick up bits and pieces of this, right? And then eventually it has it nice and organized into a good memory. But that takes time. So typically we think that this happens while we're sleeping, that this stuff gets passed on from the hippocampus to the larger cerebral cortex. So you can think of it as the cerebral cortex uploading the memory and storing it in long-term storage. And then we can access it later whenever we want to, think back. So tomorrow I can say, hey, did I post that lecture? Oh, yeah, I remember posting it at the table. That's good. If you guys have seen the movie 50 First Dates with Drew Barrymore, and I can't remember the name of the guy who's in it. Oh, Adam Sandler. Drew Barrymore and Adam Sandler. 50 First Dates is kind of exaggerating, but it's showing you an example of somebody who has a problem filing information from the hippocampus into their cerebral cortex. So she doesn't have the ability to file memories in long-term storage. So she can remember everything from like before she had an accident and damaged her brain, but she can't form new memories. So every day she'll like experience something, and then when she wakes up the next day, she doesn't remember what happened the day before. So that's what they were trying to show you, is when the transition from the hippocampus to long-term storage does not occur. Okay, so now we'll move on to the greater cerebrum, right, or the whole cerebrum in general. The cerebrum is the largest part of the brain, and it has various different high-level functions. It controls all conscious thought and intellectual functions. So all of the things that really make us human, that separate us from other animals, are up here in the cerebrum. It processes all incoming sensory information. So the sensory information that we have coming in, whether it's touch, what we see, what we hear, what we taste, what we smell, balance, all of that sensory information ultimately comes to the cerebrum and gets processed in the cerebrum. We don't sense it, we're not aware of it until it makes its way to the cerebrum. And then again, all intellectual functions, all intelligence originates in the cerebrum. The cerebrum also processes outgoing voluntary motor signals. So that's signals that are going out to our skeletal muscles, originate up in the cerebrum. We mentioned that the cerebellum monitors those signals and can adjust those signals, but the original signals that go out and tell our skeletal muscles to contract originate up in the cerebrum. Just like with the cerebellum, remember in the cerebellum we saw that the outside was gray matter and then deep inside was white matter. It's the same organization in the cerebrum. So the gray matter of the cerebrum is the more superficial area, the outer area. We refer to it as the cerebral cortex. Okay, so the outside of the cerebellum is the cerebellar cortex. The outside of the cerebrum is the cerebral cortex. And then there are various basal nuclei, like little masses that we see that get scattered throughout the deeper white matter. But most of the gray matter is in the superficial cerebral cortex. And then remember again, the significance of gray matter is that this is where all of the neuron cell bodies are located. And the white matter is where all of the axons are located. And the white matter in the cerebrum is located deep to the cerebral cortex, so it's deeper within the center of the brain. For example, like the corpus callosum. In lab, we saw the corpus callosum. That's one of the major areas of white matter in the cerebrum. The neural cortex or cerebral cortex that covers the cerebrum, that makes up the outside of the cerebrum, is highly folded. It folds in on itself, and we know that the reason it folds in on itself, so the folds increase the surface area. Okay, the more area there is, the more neurons we can put there, just the same as we saw in the cerebellum. In this case, though, we have the folds are called gyri and sulci. So the raised portions, the portions that are sticking up, are the gyri, or just one of them is a gyrus. And then the grooves, these little grooves that you see in here, those are sulci, or just one is a sulcus. Every so often, the sulci, the little grooves, are more prominent, right? They become really deep. Those deep sulci are there to divide the brain up into different lobes, right, or different sections. So we will see a few really prominent grooves or sulci, and we'll see how those help to separate the brain into different areas. So again, the sulci divide the cerebrum into 5 lobes, and these are just subdivisions of the hemispheres. The cerebral hemispheres are just the halves, right? The left half and the right half. The one lobe that's kind of weird is the insula. Insula means island. It's like the island of cortex. It's deeper than all of the other lobes. The insula is located medial or inside of the lateral sulcus. So the groove right here on the side of the brain is the lateral sulcus, and you can pull it apart, right? This is the frontal lobe here, and this is the temporal lobe. If you separate those from each other and pull them apart, you can look deeper within the brain, and that's where you'll see the insula. So the insula is just deep within, and then when you close those up, you don't see the insula anymore because it's deep, it's underneath everything else, it's its own little area. The other lobes are really easy if you remember your bones or your regions. In the front lobe is, or in the front is the frontal lobe. Then behind that is the parietal lobe back here. The occipital lobe is going to be in the very back, and then the temporal lobe again is going to be under the temporal bone. So we'll look at these in a little bit more detail. The most significant sulci, or the deeper, more prominent sulci that we look at to divide the brain up, include the 3 that you see here. The central sulcus comes like this, all the way across the hemispheres, and that divides the brain up into a frontal lobe and parietal lobe. So you see the frontal lobe is this right here, and then the parietal lobe is this area back here. The lateral sulcus is located laterally, right on the side of the brain. So the lateral sulcus, you see right here, and it's on either side. So if we flip this brain around, you looked at the other side, you'd see another lateral sulcus. The lateral sulcus separates the frontal lobe from the temporal lobe. So again, we saw the frontal lobe right here, and then the temporal lobe is over here, right, underneath the temporal bone. There's also the parieto-occipital sulcus. This is really difficult to see on the outsides of the brain. It's easier if you separate the 2 halves. So like there's this big longitudinal fissure, this big deep groove right here that goes into the brain and separates, so it goes like deep right like this, and it separates the left and right hemisphere. If we pull those apart and look on the inner edge of the brain, we can see this parieto-occipital sulcus. And what this does is it divides the parietal lobe, which is like right here, from the occipital lobe, which is in the back right here, under the occipital bone. I do have a picture with the parieto-occipital sulcus coming up in a second. So here we see all of this. First off, we're talking about the cerebrum, right? This whole thing right here up top is the cerebrum. It is really highly folded, and each of these little grooves you see like this, these are all sulci. There are a few sulci that are really large, and they divide the brain up into lobes. Right here on the side is the lateral sulcus, and then here going horizontally across the brain in the center is the central sulcus. If we were to open this up, you guys would see on the other side, like right here, going deep within the brain, there's the parieto-occipital sulcus. That divides the brain up, and we have our frontal lobe up front from the central sulcus forward. The parietal lobe goes from the central sulcus back until the parieto-occipital sulcus, and then the occipital lobe is in the very back. On the side here is the temporal lobe. This shows us the parieto-occipital sulcus. So on this one, the brain has actually been cut down the longitudinal fissure. We've separated it in half. On the inside right here, this is where you can see the parieto-occipital sulcus well, and then you just follow it around, like pretending it was a straight line. So you see right here, this slit right here, that's the sulcus, where you can really see the difference between the parietal lobe and the occipital lobe. And then also on here, this area under here, okay, where the cingulate gyrus is, the limbic lobe, that's the area where the insula is, the island of cortex. So those were the anatomical divisions of the cerebrum. Those are strictly structural. So that's just looking at the little lines and divisions, the structural parts of the brain, the cerebrum. That has nothing to do with their function. We can divide the cerebrum up based on function. So in other words, we have like a primary visual cortex, primary auditory cortex, premotor area, right? We can also divide the brain up based on function, like what each part does. So that's what we're going to look at now, right? We just saw the structural divisions, like the lobes. Now we're going to look at the functional divisions. First, it's important to note that each cerebral hemisphere, so like each side, each half of the brain, receives sensory information from and sends motor commands to the opposite side of the body. This is called hemispheric lateralization. Okay, so what this means is that the left hemisphere, the left side of the brain, the left cerebrum, sends commands to muscles on the right side of the body. Then that would be the opposite too. The right side sends commands out to muscles on the left. And then sensory information as well. So the left hemisphere gets sensory info from the right side of the body. This is important clinically because if there's damage to one side of the brain, you're going to expect the symptoms to be on the opposite side of the body. So say, and I do want you to understand this example I'm giving you right now. Say a patient has a stroke, right? The blood flow is blocked in part of their brain and part of their brain dies. So say that the part of their brain that controls muscles dies on the right side of the brain. Okay, what side of their body would you expect to be paralyzed? The left, right? The left side of the body. Say somebody comes in and they cannot feel it when you touch right here on their arm. Right? You touch their arm and they can't feel a thing. They can feel it on the left, but they cannot feel it on the right. What side of the brain would you be looking for problems? If they can't feel the right side of their body, I would be looking for a problem on the left side of their brain. Okay, so it's opposite, hemispheric lateralization. Also, keep in mind that the correspondence between a specific function and a specific region of the cortex is not precise. So we might point to, a little area right here and say that's the primary auditory area. But that doesn't mean that exactly where we draw a line, it stops at exactly that point. And 1 millimeter down, it stops dealing with that any longer. It's variable. And also the brain can change. The brain is what we call plastic, or it displays plasticity. When something displays plasticity, it can change depending on the needs. So if you damage the part of your brain that has to do with recognizing faces, the other areas of your brain can learn to compensate, and they can learn that function. Now, this takes time, but with practice, another area of the brain can learn to recognize faces. So now when you recognize someone's face, a completely different part of the brain might light up because the original part was damaged. Okay, so the brain can be variable from person to person, and the brain is really good at learning what it needs to do to survive. We're going to take a second to look inside the cerebrum, deep within the cerebrum, to look at the white matter. Remember that the significance of white matter is that white matter is where we have axons. And the axons, the neurons are out in the gray matter, right? And the axon stretches out from the neuron and it goes to another area of the brain, right? So the messages that are being sent from one part of the brain to another are traveling along axons. So the white matter is just where information is being sent, right? Like the little highways for signals to go from one cell to the next, one neuron to the next. There are three general categories of white matter in the cerebrum. We have association fibers, commissural fibers, and projection fibers. Association fibers are typically the smallest. Association fibers are connections within one hemisphere of the brain. So they don't leave the hemisphere. So association fibers could be completely in the left hemisphere or completely in the right hemisphere, but they don't leave that cerebral hemisphere. Now there are two different types of association fibers: arcuate fibers and longitudinal fasciculi. Both of these are association. So that means that either one of these, arcuate and longitudinal fasciculi, they remain in one hemisphere. Okay, so they're either in the left or in the right. Arcuate fibers are really short fibers that go from one gyrus to a neighboring gyrus. Remember, a gyrus is just like the little raised area in the brain, right? So you've got like the folds. So this would be like one gyrus, this would be another gyrus, and then the little crease in between them, right? This little line in between my fingers, that's the sulci, or the sulcus. So arcuate fibers would just go from this gyrus to this one. So they're short. They just go right to the neighboring little gyrus like this. Longitudinal fasciculi are longer, little like bundles of axons that go from one lobe to another lobe in the same hemisphere, right? So longitudinal fasciculi are longer. They go from one lobe to another lobe. Again, in the same hemisphere. Commissural fibers are bands of white matter, right? Bands of axons that connect the two hemispheres to each other. So they go back and forth between the left and the right hemisphere. This is how the hemispheres can communicate with each other. The corpus callosum is the largest of these commissural fibers. Projection fibers project from the cerebrum to other areas of the brain, lower areas of the brain. These fibers go down through the diencephalon, and then they can connect to either like part of the diencephalon, like the thalamus or hypothalamus. They can go down to the brainstem, or they can go back to the cerebellum. Sometimes they'll go down the brainstem all the way to the spinal cord. But the point of projection fibers is that they connect the cerebrum with lower areas. Okay, again, like the diencephalon, the brainstem, or the cerebellum. Both of these fibers that you see here that are shown, they are both association fibers because they're in this one single hemisphere, right? We're looking at the side of the brain, so we're just looking at the left hemisphere here. The arcuate fibers, notice, are the really short ones that just go from one gyrus to the next, right? So these little ones are just hopping across the sulcus, and they're just connecting one gyrus to the gyrus next to it. The longitudinal fasciculi are longer. These are the ones that connect the lobes to each other. So like from the frontal lobe back to the parietal lobe, or the frontal lobe over to the temporal lobe, frontal lobe back to the occipital lobe. Commissural fibers are shown in red. So the corpus callosum, the anterior commissure, these are both commissural fibers. Think like to commiserate is to come together and like grumble about something. So commissural or commiserate, commissural, come together. They connect the two hemispheres together from the left hemisphere to the right hemisphere. And then projection fibers, again, project down to another part of the brain. They leave the cerebrum. So here you see these fibers projecting down. They can go down to the diencephalon, down deeper to the brainstem, or back here to the cerebellum. All right, so again, we saw that we can divide the cerebrum up structurally into different lobes, and we said that we can also divide the cerebrum up functionally. And again, the way we divide it up functionally is by looking at what part of the brain receives what types of signals. So, and we can monitor the brain very easily and have somebody do something, and we can see what part of the brain lights up when they do it. Also, we can kind of figure this out by looking at when you damage a certain part of the brain, what does that person lose, right? What can they not do anymore? Can they not recognize faces? Can they not speak? Can they not keep beat? Can they not smell? What kinds of things are they unable to do when that part is damaged? And that's how we figure out the functional areas in the brain. We'll start by looking at the prefrontal cortex. The prefrontal cortex is the anterior portion of the frontal lobe, right? Think prefrontal, like in front of the frontal. The prefrontal cortex you see lit up here in green, right? So it's a pretty dominant anterior front portion of the frontal lobe. The prefrontal cortex allows us to behave in socially appropriate ways, ways that humans would typically behave. It allows us to decide right from wrong, to plan our behaviors, to execute our behaviors, and then it also establishes our personality. Your personality is essentially how you react to the world around you, right? Your general, natural way of interacting with the world. Again, the prefrontal cortex is only well developed in humans. It's only developed at all in primates, and it's well developed in humans. Again, we're the only animals that behave the way that we do, and that's because of our prefrontal cortex. So if you walk into a classroom, you know what to do in that classroom. You would never go into a classroom with a beer and start, dancing like you were in a club drinking a beer. You wouldn't. That's not appropriate. If you went into a funeral, you wouldn't go into a funeral talking on your cell phone really loud, cracking up, laughing and playing TikTok videos. Like, that's not appropriate. You wouldn't do that. And you don't even think about this. You just know better because of your prefrontal cortex. If we move back from the prefrontal cortex, we get to the motor areas of the brain, and then back further than that, we'll get to the sensory areas. So Directly behind the prefrontal cortex are the motor areas. We have the premotor area and the primary motor area, or primary motor cortex. You'll see me go back and forth between saying cortex and area. It doesn't matter. You can say either. So for example, right here I have primary motor cortex. You could call that the primary motor area. Doesn't matter. It's fine. It's just a certain part of the cortex. Real quick, when I just converted this PowerPoint, some of the pictures and stuff moved around a little bit. So like, don't worry about the labels that you see pre-printed. I'm going to show you the specific areas on this brain. Also, I want you guys to really realize the orientation of this brain that you see down here. It's flip-flopped from what we were looking at. So like, this is the back of the brain right here where the cerebellum is, and then the front of the brain is up over here. So we just talked about the prefrontal cortex. The prefrontal cortex, where we have our personality and behavior, was right here. Right now we're moving back from that prefrontal cortex. And the areas we're going to talk about right now are these areas in red and green. Okay, so I'm going to use the colors. I'm going to write in the color that it is on the picture here. So the motor areas. The motor areas are the areas that are anterior to the central sulcus. So in front of the central sulcus and behind the prefrontal cortex. Okay, again, the areas in red and green on this brain. The primary motor cortex is located in the precentral gyrus of the frontal lobe. So if you guys remember that the central sulcus is this line right here that goes between the frontal lobe and the parietal lobe. That's the central sulcus. In front of the central sulcus, the gyrus, the raised area, is the precentral gyrus because it's precentral, right? It's in front of the central sulcus. This gyrus right here in yellow that's right behind it is the postcentral gyrus because it's right behind the central sulcus. It's postcentral. We're talking about the red part right now, right? We're talking about the precentral gyrus that's just in front of the central sulcus. This precentral gyrus is where the primary motor cortex is located. So calling it the precentral gyrus, that would be its structural name. The primary motor cortex would be its functional name. So the primary motor cortex is this area in red right here. That whole gyrus in front of the central sulcus is called the primary motor cortex. This is the area that actually sends the signals out to our muscles to tell them to contract. Okay, the signals go down through the brain stem, down the spinal cord, and then out to our skeletal muscles. Okay, so it initiates all voluntary movements, again, of our skeletal muscles. So if you're sending a signal out to your leg to tell your leg to move, that signal started up in your primary motor cortex. Okay, the neuron is located in the primary motor cortex and it sends the signal down, goes down and tells your leg to contract. The area directly in front of the primary motor cortex is called the motor association area or premotor area. Okay, premotor because it's in front of the motor area. Also, you could think premotor because it comes first. What happens in the premotor area is that we plan movements. This is where we say, okay, you know, what muscle is going to contract, in what order are they going to contract, how much tension do they need to contract with, Movements are very complex. It's not a matter of just sending one signal out to one muscle. There's a whole plan that's involved. So the plan is made in the motor association area, and then the plan gets sent to the primary motor cortex. Then the primary motor cortex sends the actual signal out to tell the muscle to contract. This premotor area is located just anterior to the primary cortex. So all this that you see in green here, that's the premotor area or the motor association area. If we move back from the motor areas, we get to the sensory areas. There are numerous different sensory areas of the brain. Different areas deal with different senses. When we look at the sensory areas, most sensory areas are going to have a primary cortex and an association area. Kind of like we saw with muscles, right? There were two areas that were involved. We have two areas that are involved with most of our senses. The primary cortex is going to be the area where the input is first received, right? Where the sense information is first received. So from your eyes, for example, where the information is first coming into your brain, that's the primary cortex. The association area is the area where we interpret the information, where we make sense of it. So it's not just raw data, it's something that we understand. So what we're going to be doing is moving back from the central sulcus. Okay, the sensory areas are back behind the central sulcus. We're moving backwards from the motor areas. Remember we said that just behind the central sulcus, that raised area there was called the post-central gyrus, right? Because the post-central, it's right behind the central sulcus. This post-central gyrus in the parietal lobe is called the primary somatosensory cortex. It receives somatic sensory information. We have multiple different types of senses, and our senses can be grouped into somatic senses and special senses. Somatic refers to the body, right? Like soma means body. So somatic senses are senses that we feel all over the body, touch senses, right? So touch, pressure, pain, vibration, temperature, tickle, scratch, all of these feelings, anything you could feel when something touches your skin, those are somatic senses. So the somatosensory information gets sent directly into the primary somatosensory cortex. That's just raw data from the neurons. Then directly behind that, we have the somatosensory association area. These signals get sent from the primary cortex back to the association area, and that's where we interpret what we're feeling, right? You might feel, pressure in this area, pressure here, gliding here, right? Raw data that gets sent to the primary cortex. Then in the association area, you say, I'm getting a massage with deep pressure on my upper back, right? You make sense of what you're feeling. Those were the somatosenses, right? The touch senses are somatic senses. The special senses are the senses that are associated with the major sense organs in the head. So vision, right, associated with the eyes. Hearing and balance are both associated with the ear. Smell associated with olfactory receptors in the nose, and then taste is associated with gustatory receptors in our taste buds on the tongue. I'll mention all of those terms again as we go through these. So these are all of our special senses. and each one's associated with a certain area in the brain. The primary visual cortex is located in the very, very back of the occipital lobe. So the very back of the brain is the primary visual cortex. This is a primary cortex, so it receives raw data from our sight receptors in the eyes, right, which gives us vision. The visual association area is just in front of, and really it kind of wraps around the visual cortex. So the way it looks is it's like we have the primary visual cortex in the very back of the brain, and then the vision association area kind of goes around it like that. The visual association area is where we identify and understand the things that we're seeing. Okay, so like I can look right now outside, it happens to be pouring. So I see, darkness, I see these little drops, coming down from the sky. I see a flash of bright yellow. That's just raw data. Okay, that goes into the visual, the primary visual cortex. The visual association area is where I say it's raining outside, right? That's where I actually get the fact that, oh, that's water falling from the sky, that bright flash, oh, that's lightning, right? Where you can identify this stuff and make sense of it. That's the association area. The primary auditory cortex is located at the very top gyrus of the temporal lobe. So it kind of makes sense. Auditory is hearing, and it's located on the temporal lobe right here by the ears. Okay, so I always remember visual from the eyes goes straight back to the occipital lobe. Auditory for the ears just goes into the temporal lobe. The primary auditory cortex receives raw information from sound receptors. The auditory association area is just kind of behind and below the cortex, and that receives information, or that allows us to identify and understand the things that we're hearing. So if you're listening to music, just the raw sound, the loudness, the pitch, all of that, the raw information is coming into the cortex. And then the association area allows you to identify, oh, that's a trumpet I'm hearing. I recognize this song. I recognize this music. The primary olfactory cortex. Olfaction is smell. So the primary olfactory cortex is where we get information from odor receptors in the nose. This is, again, what gives us our sense of smell. The primary olfactory cortex is located on the medial temporal lobe. So this is really hard to see from outside the brain. You have to kind of like open up the lateral sulcus and look inside the temporal lobe to see the primary olfactory cortex. The primary gustatory cortex is also located down deeper in the brain. It's located in the insula, right? So both of these are located deep in the brain. And gustation is taste. So the primary gustatory cortex receives information from taste receptors on the tongue. Right? This allows us to say, is the food salty, sweet, spicy, sour, umami, which is the meat flavor. There's receptors for water, if you believe that water is flavor. But this raw data about what type of receptor is being stimulated goes to this primary gustatory cortex. Okay. Notice I didn't talk about an association area for either smell or taste. The reason for that is they don't have their own association area. There's actually one association area that, that makes sense of both olfaction and gustation. So both smell and taste are processed in the same association area. There's something called the orbitofrontal cortex. That's just because of its location in the frontal lobe above the orbit, right above the eye. And this orbital frontal cortex in the frontal lobe, again, is the association area for both smell and taste. This allows you to interpret smell and taste, and both of these things go hand in hand. They go together. Hence, when you smell something, when you walk into a room and you smell something that smells so good, you can almost taste it, right? Or the trick that kids use. When something tastes really bad, but your parents make you eat it anyways, kids will plug their nose while they eat it. That actually helps. If you eliminate the smell, that helps to tone down the overall interpretation of the food, because both of those are related so closely to each other. Here we can see all of these sensory regions of the brain. In the very beginning, we talked about the prefrontal cortex, right, which is in the very front where we have personality. In front of here's our central sulcus. In front of the central sulcus, we have motor areas. The primary motor cortex is this whole gyrus right here. And the motor association area is the large region in front of it where we plan movements. Back behind the central sulcus, we have sensory functions. The primary somatosensory cortex is the postcentral gyrus right here. That's where we receive information about touch. And then back behind it is the somatosensory association area. That's where we make sense, process, understand the things that we're feeling. In the very back of the brain, in the occipital lobe, this is where we have vision. The primary visual cortex is located at the very back of the occipital lobe, and then the visual association area kind of wraps around it. The side of the brain in the temporal lobe, this is where we have hearing. So you'll see the primary auditory cortex is right here, the very top, the temporal lobe, and then kind of back behind it and underneath it, we have the auditory association area. We mentioned that the gustatory cortex and olfactory cortex are deeper within the brain, so you have to kind of open it up, right? You open up this lateral sulcus right here so we can see deeper in the brain. You see the olfactory cortex right here on the medial side of the temporal lobe, and you see the gustatory cortex deeper within the insula on the inner lobe of the brain. The general interpretive area, also called Wernicke's area, is an integrative center that's only found in one hemisphere of the brain. Typically, it's found in the left hemisphere, so usually in the left, but not always. Wernicke's area, or the general interpretive area, receives information from all of the sensory association areas, and it coordinates all of this information to help us have access to these complex visual and auditory memories. Because we have vision processed in one area and hearing in another, and taste in another, and touch in another, and there's all these different areas, but really we have one integrated response to what's happening. And the reason for that is that this general interpretive area allows us to combine all of this information to give a complex memory, which remember we know is processed by the hippocampus and then filed in the cerebrum in general. Another integrative area that's important is called Broca's area. Broca's area, or the speech center, is also present only in one hemisphere, and again, this is typically in the left hemisphere, not always, but typically. Broca's area, or the speech center, is also associated with the general interpretive area, but its major function is to coordinate all complex vocalization functions. Hence, we call it the speech center. Speech is very difficult. Speech involves a lot of integrated things, right? We're pushing air out of the body at a specific rate. The muscles, there are a lot of muscles that are involved in speech. One, just moving the arytenoid cartilages pulls on our vocal cords, and this allows us to produce sound and to alter the pitch of our sound. But that's raw sound. Actually speaking involves the movement of a lot of different muscles in the tongue, in the cheeks, in the neck, face. There's all different muscles that are involved in producing speech. These complex vocalizations, again, are coordinated by Broca's area, or the speech center. Here, you guys see the speech center right here. Again, typically in the left, at the very bottom of the frontal lobe, just above the lateral sulcus. And then the back of the lateral sulcus, you see the general interpretive area. All right, that finishes the whole semester, guys. Good job, yay. If you have any questions, you can post a message or shoot me an email. And good job, study hard. I am confident that you will do well on this next exam.